Sometimes viruses cause a latent or dormant infection. As an example, we can use viruses that belong to a group of herpes virus family. For example, herpes simplex virus usually causes cold sores or fever blisters, and varicella zoster virus. It causes chickenpox and then later shingles. So let's talk about what is going to happen when we get infected with, let's say, herpes simplex virus. First, this virus causes a primary infection. Primary infection produces symptoms of infectious disease. For this case, for example, low-grade fever, symptoms of intoxication, which are headache, muscle ache, malaise, tiredness, and then cold sores on the surface of the mucous membranes or skin. And then patient recovers from the infection, and the virus becomes latent, and the patient is symptomless. During the latent period, herpes simplex virus will stay in trigeminal ganglia of that patient, but it can be reactivated anytime. When the virus is reactivated by a fever, for example, or cold, too much of sun or stress, painful blisters or cold sores appear on the lips once again. Varicella zoster virus causes chickenpox as a primary infection, followed by a latent infection. We call it shingles. Those are painful blistering of a strip of skin. Next group of the viruses we're going to talk about is retroviruses. An example of retroviruses will be HIV or human immunodeficiency virus. Why do we call this group of viruses retroviruses? Retro from Latin means backwards. Retroviruses use very unusual pattern of replication. For example, all cellular organisms use a DNA molecule as a template when they go through replication to make RNA copy of DNA molecule. The retroviruses use RNA molecule as a template to make a DNA copy. This process is called reverse transcription, and to perform reverse transcription, retroviruses use enzyme. It's called reverse transcriptase. Yes. So the question is, why retroviruses use RNA molecule as a template? Because retroviruses, for example, HIV virus, are RNA viruses. But why do they make DNA copy of their RNA molecule? Because remember, HIV virus uses human cells as a host cell, and our cells have double-stranded DNA. This is where all genetic information is written down. So in order for HIV virus to incorporate its genetic information into our DNA, they have to create double-stranded DNA copy of their RNA molecule. Before we continue with reverse transcription, let's talk about structure of HIV virus. As you see, this virus is an enveloped virus. In the center of each HIV virus, there are two copies of RNA molecule. So this is where all genetic information of HIV virus is stored. And then nucleic acid is covered with capsid, which is made of proteins. On the top of the capsid, HIV virus has an envelope. And as you see, there are a lot of spikes protruded from the surface of the envelope. Also, you have to remember, but there are three enzymes in the structure of HIV virus. The first one you already know, it's called reverse transcriptase. It participates in reverse transcription and makes DNA copy of RNA molecule. The second enzyme is integrase. Integrase integrates DNA copy into the host chromosome. It makes it pro-virus. And the last enzyme is called protease. Protease participate in maturation of HIV virus. It makes that virus mature. Why do we have to remember about those enzymes? Because anti-HIV drugs that we use today to manage AIDS or HIV infection actually affect enzymes of this virus. Alright, let's get back to reverse transcription. So on the first stage, RNA molecule is used as a template, and with the help of enzyme reverse transcriptase, single-stranded complementary copy of DNA molecule will be made. Next, single-stranded DNA will be copied again or duplicated, and as a result, the end product of reverse transcription will be double-stranded DNA. And after that, enzyme integrase will integrate this double-stranded DNA copy into host chromosome. This slide shows you stages of HIV replication. The first stage is called attachment, when HIV virus attaches to the surface of the host cell. After that, nucleic acid of HIV virus in form of RNA molecule will be inserted into cytoplasm of the host cell and will go through reverse transcription. As you remember, the final product of reverse transcription is double-stranded DNA molecule. It's a complementary copy of RNA molecule. Then, with the help of enzyme integrase, that DNA copy will be integrated into the host chromosome. So that means the host cell from that moment will be turned into pro-virus and will start making parts of HIV virus. Then those parts of the HIV virus will be assembled into almost mature virus, and then new virus will leave its host cell. Please remember, when HIV virus leaves its host cell, it's not entirely mature. It finishes its maturation outside of the host cell with enzyme protease. Protease enzyme makes HIV virus mature outside of the host cell. By the way, what kind of cells will be used by HIV virus as a host cell? HIV virus uses T helpers as a host cell. And finally, let's go through a little bit of history of HIV infection. Scientists believe that the first cases of HIV infection go back into 1981, when young homosexual men developed pneumocystic pneumonia and Kaposi's sarcoma. Guys, please remember, patients with HIV infection actually do not die from infection. They die from complications. Why? Because HIV virus completely destroys immune system of those patients. So basically any type of infection can be deadly for that group of patients. And most of the times they die either from a general fungal infection, and here's an example: pneumocystis pneumonia. It is a pneumonia caused by fungus. Or they develop different types of cancers, and Kaposi's sarcoma would be one of those examples. And in 1983, scientists discovered HIV virus, described it, and already knew that this virus uses T lymphocytes as host cells, and basically this virus destroys immune system of our patients. Today, scientists believe that HIV virus originated from a mutated African virus that actually affects monkeys. And for monkeys, this virus is completely benign virus. It's not a deadly, life-threatening virus, but somehow it's mutated back then and began to infect humans, causing what we know today as HIV infection that comes with AIDS. But also, as you see from this slide, scientists believe that actually the first cases of HIV infection go back into 1970s. Influenza virus is next. Influenza virus belongs to family of Orthomyxaviridae. You have to know three types of influenza virus: type A, type B, and type C. Type A has extremely wide host range. What does that mean? It means type A can infect pretty much everything: humans, animals, birds, reptiles. Types B and C infect humans only. Types A and B cause epidemics every year, and type C usually causes mild cold-like infections. Type B usually causes outbreaks every 2 or 3 years. And because type A has a wide host range, it actually able to cause what we call pandemics. Guys, a pandemics means worldwide epidemics. Structure of influenza virus. An influenza virus is a RNA virus. It has 8 copies of RNA molecules in its core. It has an envelope. It's enveloped virus, and there are spikes that protrude from the surface of that envelope. And you have to remember that there are surface proteins on the top of those spikes. Those are HA and NA proteins. Why do we have to remember about those surface proteins? Because when virus mutates, it usually changes surface proteins. There are 2 types of mutations that can be performed by influenza virus. The first one is called antigenic drift. Antigenic drift is a mild or gradual mutation. It usually happens every year, every season. As a result of antigenic drift, we have the same virus but different strand. So why do influenza viruses have to mutate? Because once infected by a strain of influenza virus, humans become immune to this specific strain. So influenza virus constantly changes. And this is what we call antigenic drift type of mutation. As I said, those are gradual or mild mutation. Second type of mutation is called antigenic shift. Antigenic shift means major mutations. As a result of antigenic shift, a completely new virus will be formed. Antigenic shift sometimes produces highly dangerous strains that cause deadly pandemics. Please remember, only type A influenza virus can perform antigenic shift type of mutation. Why? Because remember, it has wide host range. So basically, when influenza virus that infects birds get incorporated with influenza virus that infects humans, they get together, go through major changes, and eventually they create completely, or they form completely new virus. Usually this new virus is very aggressive, extremely aggressive, and causes a lot of complications in our patients. So let's repeat it once again. Antigenic drift is a mild, gradual mutation, usually happens every season. As a result of this mutation, a new strain of the same virus is formed. Antigenic shift is a major mutation, and only type A influenza virus can perform this type, and as a result of antigenic shift type of mutation, completely new virus is formed. It can be very aggressive virus, and therefore it causes pandemics or worldwide epidemics. Few examples of pandemics there are on this slide. For example, swine flu in 1918, also it's known as a Spanish flu, caused 500,000 deaths in the United States and 20 million worldwide. And actually, that number of people are killed just within basically 3 or 4 months. Asian flu in 1957 caused 70,000 deaths in the United States. Hong Kong flu in 1968, 34,000 deaths in the United States. Because some viruses are able to cause tumors in humans, let's talk about tumors. So what do you call tumors? Tumors, those are uncontrolled growth of a new tissue. Tumors can be benign, means not life-threatening. They can be invasive, means tumors grow into neighboring tissues or organs. And they can be malignant or deadly. And of course, we call them cancer. So, what does cause tumors in humans? Tumors result from unstrained growth of cells, which occurs as a consequence of genetic change. The number of ways this can happen is limited. A gene that causes a tumor is called an oncogene. Oncogenes are derived through mutations from normal genes called proto-oncogenes. Usually, proto-oncogenes regulate normal cellular division. But when they become oncogenes, however, normal regulation is destroyed and uncontrolled cancerous growth may result. Basically, proto-oncogenes are supposed to work as a gas and brake pedal, and this way they regulate cellular division. When it's time for cells to start dividing, proto-oncogenes send them a signal that it is time to start dividing. When it's time to stop dividing, they send to the same cells a signal that it's time to stop dividing. But sometimes gas pedal works and the brake pedal doesn't, and then those cells continue dividing uncontrollably. A second type of genetic change that can also result in formation of a tumor is loss or destruction of tumor suppressor gene. Tumor suppressor genes work as tumor suppressors or growth suppressors. When they stop working, then patients develop tumors. But besides the fact that something can happen, mutation of our genes, normal genes, and that eventually leads to tumor, you have to remember that some viruses also can induce tumors. The most common example, of course, will be human papilloma virus or HPV virus. You probably know it can cause cervical cancer in women. Also, another example would be hepatitis B virus. It can cause liver cancer. So the question is why some viruses can cause tumors. Because remember how viruses replicate. They have to replicate their nucleic acid into our chromosome. When they insert their nucleic acid in our chromosome, they of course can cause changes or mutations in our genes. And final slide of this lecture gives you information about prions. What are those prions or prions? Prions are infectious proteins, pieces of infectious proteins. Please remember, prions are not viruses. They have no structure of the viruses. They have no core, they have no capsid or envelope. Those are just pieces of infectious proteins, and those infectious proteins can infect humans and animals and cause very serious deadly form of meningitis. We call it spongiform encephalopathy. Spongiform encephalopathy. We're going to talk about some of those infections that are caused by prions in a little bit more details in our future lectures. But for today, you have to just remember that prions are pieces of infectious proteins that can cause very serious brain infections.